Chapter 20. Reproduction. Asexual and sexual reproduction is necessary for the identity species to exist. For all biological entities, organisms, are destined to die. While the bodies come and go, the identity persists. That is why reproduction is one of the essential features of prolonged biological existence, the law of replacement. Simplistically, reproduction is the formation of a fresh, viable, and new entity with the same identity as the parent. Offspring replace the roles of carrying the legacy of the species' identity over time. It is a general notion that sexual reproduction increases the variation upon which natural selection works, increasing the species' adaptability. Since schooling, we have been learning that sexual reproduction is to increase variability. For a long time, however, I had rebellious ideas, such as sexual reproduction is not for increasing variation, but to reduce the variation in the population as a whole. I found a good match after Googling my proposition and browsing a lot. atheist.nature.com/Citables topic page Sexual Reproduction and the Evolution of Sex 824. It is by Professor P. Otto, who, using mathematical models, describes the link between sexual reproduction and recombination, both of which reduce variation in populations. In this chapter, we shall discuss reproduction and aim to disentangle sexual reproduction and adaptation. Asexual reproduction. Asexual reproduction involves a single organism producing offspring without the involvement of gametes, sperm, and egg. This method is common in bacteria, fungi, plants, and animals. Binary fission, the simplest form of asexual reproduction, occurs when a parent cell divides into two identical daughter cells, as seen in bacteria. Budding, where a new organism develops from a growth on the parent, is seen in species like yeast and hydras. Fragmentation is another method observed in organisms like starfish and flatworms, where an organism is divided into pieces, each capable of growing into a new individual. Parthenogenesis involves the development of offspring from unfertilized eggs. While it is relatively rare, it occurs in some insects, reptiles, and certain fish species. Asexual reproduction has the advantage of rapid reproduction with minimal costs. Sexual reproduction, on the other hand, requires the fusion of male and female gametes, leading to genetically unique offspring. This process is typical in many multicellular organisms, including animals, plants, and fungi. Sexual reproduction generally involves complex mating behaviors and strategies, including internal fertilization, as seen in mammals and reptiles, where sperm fertilizes eggs within the female reproductive tract, and external fertilization, where gametes meet outside the body, as in many fish and amphibians. Sexual reproduction is generally associated, in textbooks and general knowledge, with greater genetic variation, which enhances a population's adaptability and survival in changing environments. Supposedly, it also allows for combining beneficial traits and eliminating harmful mutations, fostering evolutionary advantages. Birds do it and bees do it. Indeed, researchers estimate that over 99.9% of eukaryotes reproduce sexually. What then are the true costs and benefits of sex? Sarah P. Otto, August Weismann on sexual reproduction. The works of Charles Darwin, Gregor Mendel, and August Weismann collectively form the foundation of modern evolutionary biology by addressing complementary aspects of heredity, variation, and natural selection. Darwin's theory of evolution by natural selection, introduced in On the Origin of Species, 1859, established that species evolve over time through differential survival and reproduction of individuals better adapted to their environment. However, Darwin lacked a mechanistic explanation for how traits were inherited and how variation was generated. One of Darwin's contemporaries, Gregor Mendel, showed that traits are passed from parents to offspring in predictable patterns through discrete units, later termed genes. Although initially overlooked, his work was rediscovered around 1900, bridging the gap in Darwin's theory by explaining how variation is maintained across generations. Weismann expanded on these ideas by demonstrating the significance of meiosis and the separation of germ and somatic cells, known as the Weismann barrier. The notion that sexual reproduction increases variation, which increases adaptability, comes from Weismann's words. The communication of fresh ides to the germplasm implies an augmentation of the variational tendencies and thus an increase of the power of adaptation. He proposed that sexual reproduction generates genetic variation through recombination, a critical component for evolution by natural selection. Weismann is widely recognized as the first to articulate this concept in the context of evolutionary theory. He argued that the recombination of genetic material during sexual reproduction enhances a population's adaptability by creating diverse phenotypes, some of which may be better suited to survive and reproduce under changing environmental conditions. Germplasm and somatoplasm partitioning. We shall ask the fundamental question: what is the need for partitioning the body into germplasm and somatoplasm? The reproductive functions are confined to specific cells known as germ cells, distinct from the rest of the somatic cells. Germplasm encompasses the genetic material passed on to the next generation, typically through sexual reproduction. Firstly, it allows for the specialization and differentiation of cells within an organism. Somatic cells can undergo diverse developmental pathways to form tissues and organs specialized for various functions. The function of the somatoplasm, the vegetative body, is to harbor the germplasm and assist in making a new being, offspring. Somatoplasm navigates life's hardships, finding a mate and engaging in mating. When we speak of fitness, we speak of the ability of the somatoplasm to the most extent. Secondly, this partitioning ensures the protection of genetic material. Confining reproductive functions to a specific cell lineage minimizes the risk of mutations and alterations in the germplasm. This separation contributes to the stability and fidelity of genetic information to subsequent generations. As we discussed in an earlier chapter, the number of replications is proportional to mutations, while all else is constant. The defining feature of germplasm is the low number of replications and increased quiescence compared to somatoplasm. The fewer replications will reduce spontaneous mutations, and quiescence will reduce DNA damage due to metabolic byproducts that might lead to mutations. In humans, the minimal number of replications required for primordial germ cells, PGCs, in a female to form new PGCs in the next generation is approximately 20 divisions, around 20 during embryogenesis and oogenesis, followed by 1 meiotic division in the female and 2 in the subsequent zygote's early development. This includes the mitotic divisions during embryogenesis, meiosis to form oocytes, and subsequent fertilization and embryonic development, leading to the establishment of PGCs in the next generation. Also, the germplasm has a high expression of genes involved in genetic repair, which is likely to repair any mismatches or damage that may have occurred. Both these mechanisms then maintain the genetic integrity and reduce variation. Sex partitioning. Typically, 2 sexes are in a species solely based on which type of gamete is produced. There are variations in how sex is distributed among the members of a species, which we shall look into briefly below. 1. Hermaphroditism, a reproductive strategy where an individual has both male and female reproductive organs. Simultaneous hermaphrodites, both male and female gametes, are produced via meiosis within the same individual with homologous recombination. Sequential hermaphrodites, an individual alternates between male and female reproductive roles. Meiosis occurs during the active sex stage, contributing to genetic diversity. 2. Dioecy, a reproductive system where individual organisms are distinctly male or female. Gametogenesis involves meiosis and homologous recombination, producing genetically varied gametes that enhance the adaptability of the species. 3. Gynodoecy, a reproductive system with coexisting female and hermaphrodite individuals. Both types undergo meiosis with homologous recombination, which generates genetic variation, aiding in species resilience and adaptability. 4. Androdoecy, a rare reproductive system where male and hermaphrodite individuals coexist. Meiosis and homologous recombination occur during gametogenesis in both sexes, ensuring genetic diversity for population adaptability. 5. Parthenogenesis, a form of asexual reproduction where offspring are produced without fertilization. Automixis. Meiosis and homologous recombination occur, but genetic diversity is limited due to haploid nucleus fusion. Apomixis. Meiosis is absent and no recombination occurs, resulting in clonal offspring. 6. Pseudohermaphroditism, a condition where an individual has reproductive anatomy that does not align with their genetic sex. Gametogenesis occurs via meiosis with homologous recombination, similar to dioecious or hermaphroditic systems, depending on the anatomy. 7. Environmentally determined sex, a reproductive strategy where environmental factors, such as temperature, determine the sex of the offspring. Meiosis and homologous recombination occur during gametogenesis, producing genetic diversity vital for adapting to changing environmental conditions. Number of sexes in a species and sex changing. In sexually reproducing species, individuals are typically classified as male or female based on the production of sperm or eggs. This binary system is predominant in both animals and humans. However, nature offers numerous exceptions that challenge this framework, revealing the diversity of reproductive strategies. For instance, hermaphroditism, seen in species like earthworms and snails, allows individuals to possess both male and female reproductive organs. Simultaneous hermaphrodites, such as earthworms, produce both gametes during reproduction, while sequential hermaphrodites, such as clownfish, switch between sexes depending on environmental or social factors. Certain fungi and plants further expand the understanding of sex determination systems. Fungi, like Neurospora, exhibit multiple mating types rather than the binary male-female distinction. These mating types, functioning similarly to sex chromosomes, allow for diverse reproductive combinations, contributing to genetic variation. In plants, species like maize display monoicy, where male and female flowers exist on the same plant, whereas dioecious plants like holly have separate male and female individuals. Neurospora can have as many as 16 or more mating types, allowing for extensive genetic recombination. Ciliates like Tetrahymena thermophila possess 7 distinct mating types, requiring compatibility for sexual reproduction. Isogamous algae, such as Chlamydomonas reinhardti, also exhibit multiple genetically determined mating types, while slime molds like Physarum polycephalum boast at least 14 different mating types. Some fish species exhibit complex sex-determining systems with more than 2 sexes. One example is the bluehead wrasse, Thalassoma bifasciatum, which has a social hierarchy where individuals can switch between male, female, or third sex, termed a terminal male, dominant male. The parrotfish species, Scarus and Chlorurus, also show a form of sequential hermaphroditism with a 3-sex system. Initially, fish are females, then they may become males, and finally, dominant males may transform into terminal males. These species demonstrate a flexible reproductive system driven by social and environmental factors. Mammals, including humans, exhibit 2 primary sexes, male and female. Males produce small, mobile sperm, while females produce larger, immobile eggs. In some mammal species, there are variations in sexual differentiation. For instance, in certain marsupials, males may exhibit external reproductive organs, while females possess pouches for carrying offspring. Humans, like most placental mammals, follow a binary sex system with males and females based on chromosomal differences, XY for males, XX for females. In animals, sex refers to biological differences between males and females, typically based on reproductive organs and roles, such as the production of sperm and eggs. Gender, however, does not have the same social or cultural significance in animals as it does in humans. While animals may exhibit behaviors related to reproduction, such as mating rituals or role differentiation, these behaviors are driven by evolutionary pressures rather than societal constructs. Gender role is a cultural or civilizational characteristic. Females bear the parenting load, which is purely biological. The rest, such as cooking, cleaning, and maintaining a house, were invented when ancient humans began to develop primitive civilizations, progressing from nomadic hunting and gathering to settling, housing, and agriculture. It is evident that physical strength, aggression, and the ability to deal with threats are men's responsibilities. The man and the woman made a home, the woman kept it, and the man protected it. The degree of sexual characteristics and behaviors in humans can vary widely due to genetics, developmental issues, environmental factors, and cultural influences. However, I believe only 2 sexes, male and female, are rooted in biological reproductive roles. The presence of specific sex chromosomes and reproductive organs determines these roles. While some individuals may not fit to these categories due to conditions like gender dysphoria or intersex traits, which require respect and empathy, these variations do not change the fundamental fact that there are only two biological sexes in humans. The existence of differences in sexual traits does not invalidate this binary distinction. Sex change in animals, 8u2.bn26pf Bo. An excellent talk by Prof. Judith Mank is a good resource for understanding this phenomenon better. Sequential hermaphroditism, where individuals change sex at specific life stages, occurs in many marine and some terrestrial animals. This phenomenon is categorized into three types. 1. Protandry. Individuals start life as males and later become females. For example, clownfish, Amphiprionidae, begin as males within a hierarchical group. The dominant individual becomes a female upon the death of the current female, ensuring the continuity of reproduction. 2. Protogeny. Individuals start as females and change to males, often triggered by social or environmental factors. Many wrasses, Labridae, a group of reef fishes, exhibit this form of sex change, particularly in harem-like social structures. 3. Bidirectional sex change. Some species can alternate between sexes multiple times. For instance, certain gobies, Gobiodon, exhibit this adaptability, switching sex depending on the availability of mates and the local population's needs. Several species defy conventional reproductive norms entirely. For instance, the bluehead wrasse, Thalassoma bifasciatum, exhibits dynamic social hierarchies where the largest female transforms into a male upon the dominant male's absence. Similarly, some coral reef fishes display remarkable plasticity where sex change is triggered by environmental stimuli rather than inherent genetic programming. In plants, certain species can alter their sexual expression based on environmental cues. Jack in the pulpit, Eracema triphyllum, a flowering plant, can shift between male and female stages depending on resource availability, becoming female when favorable for seed production conditions. One should realize that, despite the costs and conditions, diverse organisms have evolved dedicated mechanisms for sexual reproduction. Why? The paradox. Why sexual reproduction? The paradox of sexual reproduction arises from its inherent costs, such as the need for two individuals and the potential transmission of harmful mutations. Asexual reproduction, which involves only one individual, would seemingly be advantageous, yet sexual reproduction remains prevalent, especially in eukaryotes. This raises the question, why is sexual reproduction so widespread despite its apparent disadvantages? Some theories suggest that sexual reproduction, while costly, offers significant benefits. It helps prevent the accumulation of harmful mutations. The problem is with the proposition that the objective of sexual reproduction is to generate variations on which evolution can operate. Let's play the devil's advocate respectfully. Various combinations of genes from both mother and father are indeed made through meiosis. Is the recombination of genes a variation? Is variation through the combination of genes the primary purpose of sexual reproduction? Suppose the objective of sexual reproduction is to generate variation. Does it imply that the ability to vary lies within the organism as an agency? Does it mean that we should expect the rates of evolution to be much faster? If natural conditions varied drastically from generation to generation, would the species evolve into a different species? Isn't there an equal probability of bad genes combining together? If there is no selection pressure, how does sexual reproduction favor adaptation? If the said variation through sexual reproduction as proposed is important for adaptability, do asexually reproducing organisms adapt at all? Don't these organisms need the kind of variation that sexually reproducing organisms enjoy? How is variation generated in the cases of self-fertilization in many hermaphrodites because both gametes are generated from the same organism, same genome? The idea that the primary purpose of sexual reproduction is to generate variation through the recombination of genes from both parents is highly debatable and warrants closer scrutiny. While it is true that sexual reproduction involves the mixing of genetic material during meiosis, it is important to question whether this process is genuinely aimed at increasing variation or diluting variation over generations. The central role of sexual reproduction in generating variation assumes that variation itself is crucial for an organism's survival and success. Is variation by meiotic recombination of various genes required? Does this variation improve the quality of the species? Suppose an organism has demonstrated competency by virtue of the fitness conferred by its somatoplasm. Isn't that good enough proof of fitness? I think sexual reproduction's role in generating variation is often overstated. The recombination of genes might result in some genetic differences between offspring, but it also carries the risk of propagating harmful genetic combinations. This means that in some cases, the mixing of alleles could lead to the expression of deleterious recessive traits or the exacerbation of genetic disorders, especially when both parents carry similar mutations. Thus, while sexual reproduction can create variation, it is not always adaptive, and the potential for harmful genetic outcomes is equally probable as the creation of beneficial variation. Sexual reproduction incorporates meiosis, a specialized cell division reducing chromosome numbers by half, enabling the formation of haploid gametes. During meiosis, homologous recombination repairs DNA mismatches and ensures proper chromosome segregation, often enhancing genetic stability. The fusion of gametes fertilization combines genetic material, reinforcing genetic repair mechanisms and eliminating deleterious mutations. The key point in sexual reproduction is gametogenesis, during which homologous recombination between the maternal and paternal genomes takes place in the germplasm. What if Weismann was carried away with the storm of Darwin's evolution and Mendel's independent assortment? What if Weismann's reasoning for sexual reproduction is wrong? And if we have been sheeping all along? What if? Sexual reproduction is often mischaracterized as a mechanism primarily aimed at generating genetic variation. In truth, its core purpose lies in resisting variation at the population level and countering evolution to maintain a species' genomic integrity and identity. Homologous recombination, a central feature of sexual reproduction in unicellular eukaryotes, highlights this function by neutralizing deleterious mutations and restoring stability to genomes destabilized by prolonged binary fission. In organisms like Paramecium tetrarelia and Tetrahymena thermophila, sexual reproduction involves an exchange of micronuclei, with recombination ensuring the repair and maintenance of the genetic template. Similarly, in Saccharomyces cerevisiae and Schizosaccharomyces pombe, homologous recombination during meiosis corrects accumulated errors, resisting the incremental changes that would otherwise erode genomic integrity. By purging harmful mutations and repairing DNA, sexual reproduction actively resists unwanted variation that compromises survival. Sexual reproduction, rather than serving to enhance genetic diversity, functions primarily to dilute variation over generations. This process ensures genetic stability by homogenizing alleles through recombination and independent assortment, preventing the accumulation of extreme traits that could destabilize a population. By continuously reshuffling genetic material, sexual reproduction eliminates both deleterious and beneficial mutations at a population level, maintaining a balanced genomic composition that favors survival over radical change. This mechanism stabilizes the species, reducing the rate of evolutionary divergence and preserving an optimal genetic framework for existence. Large population size further strengthens this stability by buffering against genetic drift, preventing rare mutations from becoming fixed. In a small population, genetic drift can amplify deviations, but in a large, randomly mating population, recombination ensures that genetic extremes are diluted back into the collective gene pool. Random mating reinforces this homogenization by equalizing allele frequencies and preventing localized adaptations from disrupting overall genetic consistency. By moderating genetic fluctuations, sexual reproduction limits evolutionary shifts that could compromise species survival. This controlled reduction in genetic variance sustains existence by maintaining a proven genetic configuration, ensuring long-term adaptability without necessitating abrupt evolutionary changes that could lead to maladaptation or extinction. Rather than being an engine of diversity, sexual reproduction functions as a genomic safeguard, preserving the species' identity by maintaining its genetic blueprint. This perspective redefines its role, emphasizing its contribution to the continuity and stability of life. Far from promoting evolution, sexual reproduction acts as a force against unchecked variation, ensuring the existence and persistence of species across generations. Deletions of homologous recombination genes typically result in increased mutation accumulation due to the loss of recombination and DNA repair functions. In both bacteria and eukaryotes, whether unicellular or multicellular, the inability to properly repair DNA leads to genomic instability and failure to resolve DNA damage. In particular, the disruption of homologous recombination during meiosis results in chromosomal defects, genomic instability, and fertility issues. These consequences are especially notable in multicellular organisms, where impaired homologous recombination causes infertility and can lead to a range of chromosomal abnormalities. Therefore, homologous recombination is critical for maintaining genome integrity across diverse organisms. Revisiting homologous recombination. Homologous recombination, a fundamental process in meiosis and sexual reproduction, has traditionally been viewed as a mechanism to enhance genetic diversity by reshuffling alleles. This is a faulty assumption and reductionist in observation, looking only at a span of generation. Earlier scientists have assumed or reasoned the generation of a combination of alleles as if the organisms have an inherent tendency or inclination to generate variation. Instead, look at homologous recombination and sexual reproduction as a way to reverse the variations or at least dilute them. What if variation is the norm and reversing the variation is what all organisms are doing? The traditional interpretation may overlook homologous recombination's primary purpose to ensure genetic stability by minimizing harmful variation. Sexual reproduction, in this light, operates as a tool for preserving genetic integrity across generations through iterative cycles of homologous recombination. During meiosis, homologous chromosomes undergo recombination, facilitating the exchange of genetic material. The precise alignment and subsequent exchange of DNA segments are mediated by the formation of synaptonemal complexes and the repair of double-strand breaks. While the superficial outcome may appear to be the generation of novel allele combinations, the deeper biological significance lies in the ability to repair mismatches, correct deleterious mutations, and homogenize advantageous genetic configurations. Gene conversion. Over successive generations, the cumulative effect of homologous recombination, paired with selection pressures, filters out harmful mutations and preserves functional genomes. Rather than driving random genetic variation, recombination prioritizes correcting deviations from a genetic identity that has proven successful within a given environment. Consequently, sexual reproduction, through iterative recombination, refines the genome, reinforcing genetic integrity and reducing variance. The stabilizing role of homologous recombination can be observed in its ability to suppress the accumulation of deleterious alleles, Muller's ratchet, and its influence in maintaining genetic cohesion within populations. While diversity is a byproduct, the central function of sexual reproduction and recombination appears to be maintaining an optimized genetic framework. This interpretation challenges the conventional view, suggesting that sexual reproduction serves as a biological mechanism to enhance genomic fidelity over time, ensuring survival in changing environments without the uncontrolled proliferation of harmful genetic variability. Sexual reproduction as a mechanism of genetic conservation and the constraint on evolution. The emergence of phenotypes is a complex and highly regulated process, shaped by the interaction of multiple genes dispersed across the genome. Phenotypic manifestation requires the coordinated action of numerous genetic elements, yet the loss of function in even a single gene can be sufficient to abolish the trait. This genetic interdependence underscores both the stability and fragility of biological systems. In multicellular organisms, the genes responsible for a single phenotype may be located on different chromosomes, making their inheritance subject to the mechanisms of sexual reproduction. Sexual reproduction, through the process of meiosis, serves two primary functions. First, it reshuffles chromosomes, ensuring that offspring inherit a unique combination of genetic material from their parents. Second, meiotic recombination facilitates the mixing of genes within chromosomes. While often perceived as a driver of evolutionary change, these processes introduce a fundamental constraint: the continual fragmentation of advantageous genetic combinations. Consider a scenario where two beneficial mutations emerge in an individual, one on chromosome A and another on chromosome B. If inherited together, these mutations could significantly enhance fitness. However, during meiosis, independent assortment and recombination ensure that these genes are rarely inherited together. Even if they exist on the same chromosome, crossing over may separate them. In a randomly mating large population, the probability of these beneficial mutations recombining in subsequent generations is exceedingly low. Instead of facilitating the rapid establishment of advantageous traits, sexual reproduction disperses them, preventing dramatic increases in fitness. The principles of natural selection create an asymmetry in how mutations persist within a population. Deleterious mutations that reduce fitness are swiftly eliminated through purifying selection. However, beneficial mutations, despite their potential advantages, face a greater challenge in becoming widespread. Natural selection favors their proliferation, but the disruptive forces of genetic recombination, independent assortment, and random mating act against their stable accumulation. As a result, evolution is not an unchecked force of progress. Rather, it is constrained by the very mechanisms that govern inheritance. When sexual reproduction operates within a large, randomly mating population, the probability of evolutionary change decreases, while the probability of genetic continuity, existence, is reinforced. The vast genetic diversity within such a population ensures that individual mutations, even if beneficial, are constantly mixed and diluted. Evolutionary transformations require the stable inheritance and accumulation of beneficial mutations, but this becomes increasingly improbable when recombination disrupts favorable gene combinations before they can establish dominance. In essence, sexual reproduction, rather than accelerating evolution, serves as a stabilizing force that preserves the existing genetic framework. Evolutionary acceleration occurs under specific conditions, primarily when population sizes decline drastically or when mating is highly restricted. In a small population, genetic drift plays a more significant role, allowing mutations, whether beneficial, neutral, or even slightly deleterious, to become fixed more readily. Similarly, in a population with limited mating choices, advantageous mutations have a higher probability of persisting and accumulating without being diluted. These conditions create an environment where selection can act more decisively, leading to rapid evolutionary shifts. In contrast, when population sizes are large and mating is random, the genetic landscape remains relatively stable, impeding the emergence of novel traits. In a large, randomly mating population, the forces of recombination and independent assortment serve as buffers against drastic evolutionary change, ultimately favoring the persistence of existence over rapid transformation. Evolutionary leaps occur primarily when genetic constraints are lifted, whether by population bottlenecks, reproductive isolation, or extreme environmental pressures. In the absence of such factors, life remains in a state of equilibrium, where change is possible but slow, ensuring continuity over disruption. Inbreeding and outbreeding, evolutionary implications. In academic discourse, inbreeding, characterized by mating closely related individuals within a population, can lead to several detrimental genetic consequences. When closely related individuals reproduce, the likelihood of inheriting identical alleles from both parents increases, including deleterious recessive alleles. By the same rationale, any allele with a fitness advantage can accumulate. With numerous generations, the new allele will establish its predominance within the inbreeding population, and possibly could also contribute to the dominance of this population in the niche. In that way, more alleles of increased fitness could accumulate, and this population could form a new species. Indeed, those individuals who've accumulated deleterious alleles will be eliminated by natural selection. Inbreeding, characterized by mating among closely related individuals, increases homozygosity within a population. This promotes the rapid fixation of beneficial mutations, as related individuals are more likely to carry and pass on the same genetic traits. If a beneficial mutation arises, inbreeding enhances its likelihood of becoming homozygous within just a few generations, especially in small, isolated populations. This rapid genetic uniformity can lead to the establishment of unique traits within a population, fostering divergence from other populations. Over time, this genetic divergence and ecological or behavioral isolation can result in reproductive barriers, a key requirement for speciation. Additionally, the reduced genetic diversity in inbreeding populations amplifies the effects of genetic drift, further increasing the potential for evolutionary novelty and the emergence of new species. Thus, inbreeding plays a critical role in accelerating the genetic changes necessary for speciation, driving the evolutionary process forward. Outbreeding and existence. In contrast, outbreeding, or random mating between unrelated individuals, promotes genetic mixing within a population. While outbreeding slows the fixation of specific mutations due to the dilution of alleles through genetic mixing, it enhances the population's overall fitness by reducing the likelihood of deleterious recessive traits becoming homozygous. By maintaining genetic mixing, outbreeding ensures that populations remain robust, favoring existence over rapid evolutionary change. Speciation in outbreeding populations typically requires geographic or ecological isolation to create conditions that reduce gene flow and allow divergence to occur. The rationale for inbreeding driving speciation while outbreeding prevents it lies in the mechanisms of genetic variation within populations. In smaller, isolated populations undergoing inbreeding, the restricted gene pool allows novel variations or mutations to become fixed more easily over generations. This fixation of traits, often amplified by selective pressures, can lead to reproductive isolation and the emergence of a new species. Importantly, when homologous recombination occurs in smaller pools, the predominant allele has less probability of reversing rare variants, allowing those variants to persist and contribute to speciation. This amplifies changes unique to the inbred population, enabling divergence from the ancestral species. In contrast, outbreeding introduces genetic material from a larger, more diverse population, which increases the likelihood that dominant alleles will reverse or dilute variants introduced through mutations. Homologous recombination in such cases often favors the reestablishment of the predominant alleles of the larger gene pool, maintaining genetic stability and hindering divergence. Outbreeding stabilizes the genetic structure of populations, ensuring the continuity of an existing species rather than allowing fragmentation into distinct lineages. Outbreeding in mammals is supported by specific mechanisms and behaviors that promote genetic mixing and avoid inbreeding, ensuring the greater existence of the species. 1. Kin recognition and avoidance. Many mammals, including ground squirrels, use olfactory cues to recognize relatives. This prevents mating between close kin, encouraging mating with unrelated individuals. 2. Sex bias dispersal. In species like African elephants, males typically leave their natal groups upon reaching sexual maturity. This reduces the likelihood of inbreeding and fosters gene flow between different populations. Territorial overlap in mating. Tigers are solitary but establish territories that overlap with others. Males travel across territories to seek mates, reducing inbreeding within isolated populations. 4. Dominance and roaming in polygynous systems. In gorillas, dominant silverback males often migrate to live new groups or mate with females in neighboring groups. This spreads their genes and prevents prolonged inbreeding within one troop. 5. Promiscuity in females. Female bats often engage in polyandry, mating with multiple males. This behavior facilitates sperm competition, maximizing genetic variability in their offspring. Outbreeding is favored not to prevent the accumulation of deleterious alleles, but to reverse all kinds of variations through homologous recombination, thereby infusing the existence of species and reducing new speciation. However, when the population size reduces drastically or is isolated from the rest of the population because of barriers, new species emerge over repeated inbreeding for several generations without any interference from the outside population. In the context of speciation, inbreeding and outbreeding play distinct roles, each favoring different aspects of evolutionary and existential dynamics. Inbreeding accelerates evolution, while outbreeding is more conducive to populations' sustained existence. We have discussed many things that are an addition or a contradiction to the existing models. We should remember, especially with mathematical modeling, that all the outcomes depend on the assumptions of the components, the interactions among the components, and the consequences of the interactions. We can give a mathematical expression to almost everything as long as the above factors are defined. In the end, we have to assume some aspects to generate the equations. That is a major limitation in the reliability of the models. Indeed, mathematical models are excellent tools to predict and express phenomena that are not immediately evident. The contradictions between my views and the already established views are mostly due to assumptions, and I am more than happy to be disproved. As I conceded already, my mathematical expressions are incomplete. One thing, with regard to sexual reproduction and homologous recombination, in my view, should be accounted for multiple generations, not the outcomes of one generation. The frequency of occurrence of alleles, the probability of gene conversions, the viability of gametes upon recombination, and the consequences in several generations should be accounted for to better the existing models on the relationship between sexual reproduction and evolution. All models are wrong, but some are useful. George Box. Stable existence of asexually reproducing organisms? Suppose variation is the norm and sexual reproduction via homologous recombination is to reverse variations. How could an asexually reproducing organism be a stable species? Firstly, we need to see if any species in several generations does not indulge in homologous recombination with genetic information from a different organism, kin, or another cell of the same body. If there is homologous recombination, it must be considered in light of reversal of change because external DNA can always provide templates for reversal. In so far of my exploration, there is no species in which homologous recombination with exogenous DNA from another cell or another organism doesn't occur. I bet there wouldn't be one. Suppose a species does not include such homologous recombination. In that case, the possibilities are that these organisms are at the mercy of stringent natural selection. Only those without variation survive and others die. They reproduce in large numbers, among which only the best fits survive and the rest die. There are three other unexplored possibilities. There is an immense reduction in errors because the DNA polymerases have high fidelity. They have multiple high-efficiency repair mechanisms to repair any damage. The cells that differentiate into reproductive cells might have participated in homologous recombination with the DNA of the adjacent cells, thereby reversing any changes that may have occurred. We have not yet noticed if these species may have some form of sexual reproduction. In the case of bacteria, as the cultures age, a form of competence emerges, because of which they are likely to take up genetic material from the environment. They have molecular mechanisms to distinguish if exogenous DNA is from their kin or a different species. A specific sequence, such as chromosome hotspot instigator, CHI, in E. coli, if present, will be processed for undergoing homologous recombination. If such a sequence is absent, the DNA is degraded and used up as an energy matter source. I assume most bacteria have mechanisms that help them retain their genetic integrity. It is conceivable that many of the bacteria have a high rate of replication, which increases the likelihood of mutations. E. coli, if growing in optimal conditions, divides every 20 minutes. If organisms have an inherent tendency to accumulate variation, sequences like CHI shouldn't have been there. Variations would be higher if DNA from different species were integrated. Scientists have attributed other mechanisms for too long to support the notion that these organisms tend to increase variation. For example, transposons. What is the use of the transposons? Again, exhaustingly, it generates variations. No, transposons are just genetic parasites. They just propagate like other propagating entities. They have no role or need to create variation in the host. Just that variation happens.